Hello, my name is Jacquelyn Lee. I'm a field scientist with Dow Agrosciences. I currently do research on the Sentricon colony elimination system. Today I would like to talk to you about DNA fingerprinting. DNA fingerprinting is the latest technology for termite colony ID. It's very important for us to be able to identify termite colonies in our research. We really want to prove how many colonies we have around a structure within 1 year and how many of those have been effectively eliminated. So I'm going to go into the different procedures that we go through when we use this technology for colony ID. So the very first thing we do is we have to have Sentricon installed around a structure. In this instance, we used Recruit HD. We would go and check the stations. Whenever there was a Recruit HD station that had termites feeding on the bait, we would collect 10 to 20 termites from the bait. We would take the termites, put them in an alcohol vial, and then transfer them to the lab. In the lab, we would take the termites, put them in an Eppendorf tube, which is just a little plastic tube, and we would grind the termites up, just smash them. So when you smash the termites, all of their DNA would fall out into the liquid. Once we have the DNA in the liquid, what we have to do is amplify the part of the DNA that's going to tell us who am I. Because what we're really interested in here is what colony does this termite belong to. So this DNA strand that we're looking at is very, very long. It's a very long strand in that solution. And what we have to do is pick out that little bitty piece that we're interested in. So we have these special pieces of DNA called primers. And we have a little primer for every piece we're interested in on that long strand of DNA. So we pour the primers into the solution, and they attach to that long strand of DNA, and they replicate when we put the sample into a thermocycler. Now this thermocycler regulates the temperature to make a chemical reaction that causes replication by the PCR. And this is a picture of the replication process, and we get exponential growth of our target DNA pieces. So we start out with maybe one strand and we'll end up with thousands. And it's really important that we have thousands of strands so we can visualize them, because one tiny piece of DNA is very difficult to visualize. So to visualize our DNA, we use a process called gel electrophoresis. And here's a picture of the gel electrophoresis box. And here's a picture of what is called a gel. Now, to start this process, you take that sample from this thermocycler and you pipette out the sample into the wells. And up here you can see little wells. So DNA has a very slight negative charge. This slight negative charge is a great characteristic, because we can actually use that by applying a slight electric gradient over a liquid poured into the gel box, and the DNA will migrate closer to the positive electrode. So each one of these little DNA fragments, they're different sizes, so they actually move at a different rate. So what you get is different banding patterns. The smaller the piece of DNA, the faster it moves. So the farther down it'll go. And since we looked at many different targets, we get what's called a banding pattern. So for example, in this picture, we have two different samples. A sample from, let's say, station 1, and then samples from station 2. And you can clearly see that the banding pattern is the same for the first 10 samples and the same for the second set of samples. So we clearly have two different colonies here. Now this technology is very important to us because it allows us to quickly determine if we're dealing with one or two colonies at a site. We're able to monitor how many colonies we find within a year, and through this research, we've seen multiple colonies arrive within a year. And also, we have found that multiple colonies will move in and hit a bait that has been fed upon by a different colony. So this technology has been really, really important to us to help prove that we can eliminate multiple termite colonies within 1 year. And now I would like to show a DNA precipitation demo. A really interesting characteristic about DNA is that it is insoluble in ethanol. Now this is a tube of ethanol, and I'm going to add some extracted DNA. You can see that the liquid is becoming milky as the DNA is precipitating. And this is the DNA precipitate. You can see that the DNA has formed long strands within the tube. Now this is the same DNA that we would use for this technique, but it's just in one very, very long strand. So you can see why we have to use the primers to get the little strand that we're interested in. I would like to thank Dr. Ed Vargo from NC State and his laboratory. They processed many of the samples that we used in our studies, and also Ed Vargo pioneered many of the techniques that we use for termite colony ID. And lastly, if you have any questions or concerns, please contact your local Dow Agrosciences sales representative. And thank you.